Hello and namaste. My name is Prokesi. I'm from Manipal College of Medical Science, currently studying in second year. Okay, so we'll be talking about the microbiology sporters and practicals of the first semester. Okay, so getting into the microbiology sporters. So these are the slides. These are quite important. Everything is important because these things can be placed randomly to any of the groups. So please, the answers are also given and take care of it. Okay. So this is 5% phenyl. Status use. It is used to disinfect the hospital floor. Concentration. It is obviously 5%. Okay. Now, so like this, the gram negative, the gram positive cocci, bacilli can be given, and you have to identify it based on the staining that it has taken, as well as the shape of the bacilli. If it is oval shaped or round, that's cocci, and if it is elongated, that's bacilli. Okay, and also remember some of the examples. Okay. Now, talking about the blood agar. So the main difference, you can read about the description in your books and in your manuals. Okay, but the main thing to identify it is the opacity of the agar plate. Okay, so when the agar plate is kept on the desk, or similarly in this case over the paper, you cannot see the underlying letters. So that's opaque, and that is blood agar. Okay, whereas in the mechanical agar you can see the words, so it is not much opaque. That is why from this you can identify whether that's a blood agar or it is a mechanical agar, and you can read the description. You can you have to take care of uh, what organisms can be cultured in such agar plates and what is the specific use. Like in mechanical agar, the lactose fermenters and lactose non-fermenters can be distinguished. The lactose fermenter colony will form pink color colony, whereas the lactose non-fermenter will not form any sort of colorations. That is due to the fermentation, and thus the acidity is increased, and that is why the coloration is pink. This is Ames transport media. These are for the second semesters. And this is LJ media or Lowenstein-Jensen media. You can see a slanted slope. Similarly, the colorations is quite bluish in color, and the main thing you have to identify is the the contents or constituent of this medium, that is, it contains hens egg. Similarly, malachite green, which is the specific constituent, as because the malachite green will only allow Mycobacterium tuberculosis to grow, whereas the other organisms, other bacteria won't grow. Similarly, it is sterilized by inspection. So you have to identify and know that the medium are sterilized by which method. So usually the culture medium are sterilized by autoclaving, but in case of LJ media, it is done by inspection. Okay. And this is a bacteriological wire loop. It is sterilized by heating in the Bunsen burner to red heat. You have to identify this and the use. Okay. Yes, this is also important. It is filamentous branch gram positive bacteria. You have to identify and memorize this picture as a whole. You can see the gram positive behavior, since it is quite violet in color and it is very branched in structure. So example are nocardia and streptomyces. So to identify the candida, you have to see the budding. So you can see a bud being formed here. So, and also take care that you don't mistake the candida with the gram positive cocci. The gram positive cocci are very small and there are no budding. So try to find about the cells that have the buds and they are quite large in size. So similarly, you can also see here the budding. This is microtiter ELISA plate. Know the use. This is a multi-purpose specimen container. You have to know the use and how it is sterilized. LJ medium, we talked about it. These are the description of blood agar. Why is it used? To differentiate between alpha and beta hemolysis. This is cotton swab sterilized by hot air oven. Give better emphasis on the sterilization and the use. This is Meccan-Key agar. Bacteriological wire loop again, Gram Candida. This autoclave tape, use. Candle jar. Yes, this is Conidia. Chocolate agar, it is, the color is chocolate in color, like brownish in color, so you can identify through it. This is CH filter. Know the use for the sterilization of solutions such as sera, sugar, and antibiotic solution. This is gram positive and gram negative bacilli. See the shape and the color. Septate hyphae of the mycelium. This is a nutrient agar. It is quite transparent or translucent and it has no color. Know the use. Know the constituent. Robertson cooked meat medium. You can see the picture in the Google as well. You can see there is a small bottle with meat pieces, or you can see the small pieces of meat inside the jar or the bottle. And know this, glutathione maintains the anaerobic condition and it is used for cultivation of anaerobic organisms. This is HIB tridot, which is used for the screening test for HIB. You can find the SDA agar picture in the Google. This is autoclave. You can see here as well. Know the use and the temperature as well as the pressure and the time, holding time. This gram positive cocci, as you can see, the size is very small as compared to that of Conidia. This is biphasic medium. You can see there's a solid medium on the wall of the bottle as well as a liquid medium. So there's a two phases. So biphasic medium. That's anaerobic jar. Know the use. As you can see here, the Meccan-Key agar, the lines of the table can be seen, whereas in blood agar it is quite opaque and the line cannot be seen. About the antibiotic susceptibility testing, as you can see, this is the picture. You can see similar thing in the spotters. And know about the use and about the inhibitory zones. So without any further ado, we will go into gram staining and the important questions. Okay. So about the gram staining, you should know the procedure at first. You must know how to do it. Similarly, for the VIVA, you have to know about the examples of gram positive and negative cocci and bacilli. This is a must know. Similarly, about the theory of gram staining. There are two types of theory. One is pore size theory and another is cytoplasmic theory. You can have the idea of these theories. They're written in the book as well as in the manuals. So, about the gram staining, you must know what are the primary stain. What is the primary stain? Name of the primary stain used in the gram stain, which is methyl violet. Similarly, examples of other primary stains that are crystal violet. The primary stain used is crystal violet. The other examples are crystal violet, methyl violet, similarly gentian violet, okay. And what is the mordant? It's iodine. Similarly, what is the decolorizer used? It's acetone. Similarly, alcohol can also be used, like ethanol, as well as aniline can also be used. But you must know the holding time. The acetone, it is kept for just 1 to 2 seconds and it is washed right away, whereas the alcohol, it can be kept for 20 to 30 seconds. Okay, similarly, what are the counter stains? What is the counter stain used in gram stain? That's safranin. Whereas the other examples of counter stains can be carbolphosphine, similarly neutral red. Okay, so at first you must know the names. Now you must know what are the uses of primary stain, what are the use of mordant, what are the use of decolorizer, and what are the use of counter stain. Okay, so what is the use of primary stain? It is used to stain at first, okay, and the mordant, it is just used to hold the stain. The decolorizer, it is used to, obviously the name is used to decolorize or remove the color. And counter stain, if the primary stain has been decolorized, has been removed, then the counter stain takes place on that particular organism, and the particular organism gets the counter stain as such in case of gram negative organisms. Okay, so know this. Now you must know about the gram stain and what are the other types of staining. Okay, so types of staining can be simple stain or negative stain or impregnation method or staining by impregnation method. Okay, the simple stain examples is methylene blue. While that of negative stain, it's India ink. About the impregnation method, it is silver staining, silver impregnation method. It is usually used for spirochetes. Okay. Similarly, if you answer properly, then the teacher might ask about the modification of gram stain. Okay, so it can be, one is Jensen's modification, another is Brown and brain modification, another is Kopelov and Biermann's modification. Similarly, Jensen modification, you must know what is the modification done. In this, the decolorized use is absolute alcohol and neutral red is used as counterstain, and it is used for gonococci and meningococci. Similarly, Brown's and brain modifications, it is done for actinomyces, and in Kopelov and Biermann's modification, the primary stain used is methylene violet and the counter stain used is basic fustine. You must know about the use as well. Find it out. Okay, now I'm going to talk about some of the possible VIVA questions. These are important to know and these are quite simple. And if you are not able to answer them, then the VIVA teacher would be quite disappointed on you. So don't do that. Okay, I'll just write about the topics, and these are the things you must know, and try to learn much more than this and know about much more topics, because it's all about the mood of the VIVA teachers, and they may ask anything related to the particular syllabus. Okay, so in the basic concept, know about the culture medias. It is quite important. About the culture medias. Similarly, about the growth curve of bacteria, and how the bacterial spores are formed, okay, then methods of anaerobiosis, similarly about the decontamination, disinfection and sterilization. Autoclaving and hot air oven are quite important. Similarly, the teacher might ask about what are the different types of or different methods of sterilization. It is written in your manual as well as it is present in the book. And names of normal flora, and if possible know over the sites as well. Similarly, about the antibiotic susceptibility testing, and about hospital acquired infections and management, or we can say not management, we can say how they are prevented. Okay, now The must know is bacterial genetics. It is a quite simple and a short topic, and there are many things that can be asked from the bacterial genetics. So know about it quite well. About the immunity, uh, have the basic concept about it. What are the types of immunity? Classification of immunity. And know the basic ideas about each type. Okay, know what is antigen, what are antibodies, what is mean by active immunity, what is the mechanism about the humoral immunity, and passive immunity, about local immunity, herd immunity, antigens, what are antibodies, or the type of antibodies, IgA, IgG, IgM, IgE, IgD, right? These are some of the conceptual things that can be asked. About the complement system, about the various tests that can be immunological test, like ELISA, as well as neutralization, precipitation, agglutination test, radioimmunoassay. Main is ELISA, as well as precipitation and agglutination, as well as neutralization. Okay, and hypersensitivity is quite important as well. Sorry. Or the types of hypersensitivity and the mechanism of hypersensitivity. Know this. And names of some of the immunodeficiency diseases. Okay, hypersensitivity is quite important as well. Now about autoimmunity and the mechanisms and examples of some of the autoimmune diseases. Now about immunization, or the types of vaccines, and examples. It is very important. Okay. So now we go to paper 2. In paper 2, things are quite important, and they are quite smaller topics, and like about the skin infections, what are the type of skin infections, the viral, bacterial, fungal, viral, bacterial, fungal, and parasitic. You must know about these topics. These are very important. You must know the names of such infections. You must know the pathogenesis. You must know the causative agent. read it as a whole. These are important topics. The integumentary system is a very important topic as a whole. Because many things can be asked from here. Bacillus anthracis, Mycobacterium leprae, atypical mycobacteria, the names of some of the atypical mycobacteria. Similarly, about gas gangrene, superficial mycosis, dermatophytosis, subcutaneous mycosis. Lab diagnosis is very important. About the viral infections and parasites. Okay. Now, going to MSK, you must know about osteomyelitis. It is a must know. Pathogenesis and lab diagnosis, as well as of septic arthritis. These are quite similar. These are the things, these are important topics. You must know about them, but it doesn't mean that the questions will be asked from these only. Okay, so you have to know something about all of the topics. You must be able to say something when the teacher asks, and try to remain humble and be respectful to the teachers. Okay, so read more, know more. Thank you.